I'm going to be talking about the Zuretinol chromophore replacement program that we're involved in. And this is very near and dear to my heart because I'm one of the inventors of this while I was at the University of Washington. We are a public traded company, so we have our forward-looking statement. And to understand this technology, I have to tell you a little bit about the biochemistry of the retina. And in the normal state, vitamin A is absorbed from the serum. It's enzymatically converted into 11-cis retinal, which binds opsin in the case of rods and forms rhodopsin. This then can be activated on by light, and it starts the visual transduction cascade, and you get vision. Now, if you don't have one of these enzymes that converts the vitamin A, you don't get 11-cis retinal. You can add as much vitamin A as you want, and without those enzymes, both of those enzymes together, you don't convert it to 11-cis retinal, and you lose vision. And so these people who have mutations in these are in the RPE65 or the LRAT genes, develop LCA or retinitis pigmentosa. The gene therapy folks and Spark and some others have decided to replace the RPE65 gene in the patients that have those mutations, and there's lots of data out there from them. But what we decided to do was to see if we could replace the chromophore. So instead of replacing the protein, we were going to replace the 11-cis retinal. And what we found is that 9-cis retinal works almost identically to 11-cis retinal. And it's really the only compound that we could find that binds opsin, can be activated by light, and then unbinds from the opsin. So we developed a prodrug that we'll call, that's 9-cis retinol acetate or Zuretinol Acetate. And that can be delivered orally, intravitreally, subcutaneously, really any way you deliver it, it seems to get to the eye. And we wanted to see if that would work. And so we did that, and we got transgenic mice that have these proteins knocked out. And what we found is that they would start with no ERGs, virtually no vision, and then we treat them with this orally, and by the end of the day, their vision returned. And we did this on hundreds of animals, and we were so impressed with this technology that we, like the commercial, as we actually bought the technology. We started a company and bought the technology from the University of Washington. We licensed it from them. And then we subsequently licensed the technology to QLT, sub-licensed it. So then we went ahead and with QLT and went to a larger animal. And this is the same kind of dog as Lancelot, who had gene therapy, who is blind from RPE65 mutation, naturally occurring mutation. You can see that dog really can't see very well. And then we treat with one injection in the vitreous, in the right eye, and you can see that the dog now can navigate this very sophisticated obstacle course, is happier, waving its tail. That's after 3 days after this intravitreal injection. And then after 14 days, from that one injection, the dog was much more agile and could see very well to get around these obstacles. So from there, we cured mice, we cured dogs, so we decided that we should start on humans. And so we did a phase 1b trial for both RP and LCA patients that have these mutations. We did it in, it's a multi-center trial, it was open label. We gave 7 days of oral dosing. This is, of course, after you go through all the IND-enabling studies, and in this case, healthy human volunteer studies. And what we found is that you can see on the right, here's the summary of the results, and you can just really look at the bottom, that in 81% of the patients that we treated, they responded to visual field or visual acuity, which we defined with Goldman visual field as having a visual field that expanded by at least 20%, or visual acuity that improved by at least 5 letters. And so I'm going to show you an example of one of the better patients here in the trial. So this is a patient, we have a couple of patients that responded here, and you can see that this patient had a very narrow visual field when they entered the trial. And then after 7 days of dosing, it expanded, it developed those temporal islands you see there. And then by 4 months from that 7 day of dosing, they developed what was a pretty full visual field to the 5/4E. And this particular patient also improved their visual acuity from 2250 to 2070 and no longer needed her cane to ambulate while the drug was on board. We then went and did a repeat trial where we wanted to give the same dose again. We repeated it up to 3 times, and you can see in the panel there that every time you give the dose, this is visual field area, that the visual field goes back up. So there's definitely a dose response here. And in this study, what we found is that 70% of patients were Goldman visual field responders and 70% were visual acuity responders. This is a retinoid class of drug. It is a retinoid, so we saw the typical things that you'd see with retinoids: headache, photophobia, and some mild and reversible changes in transient changes in blood serum levels. And we had 1 or 2 serious adverse events, 1 allergic reaction in the healthy volunteer study, and 1 patient that had elevated intracranial pressure that reversed with treatment. And this patient actually requested more drug because it was one of the good responders, but we didn't treat them again. And 144 people have been treated to date with this drug. So what's next? I was hoping to be able to tell you we started a phase 3 trial, but we are just about to start a phase 3 trial. It's going to be placebo-controlled, double-masked. It's a multi-centered in several continents, and we plan to start this very soon. So why bother? We have Spark out there and these other companies looking at the same mutation, the RPE65 mutations. And I think that there's still value for us to keep moving. We have potential for more tools in the doctor's toolbox. That's always good for the doctor. This is oral delivery, so no surgery is necessary. The drug's delivered to the entire retina of both eyes. And the data suggests that it's reversible on effects of cessation. And there may be some merit in combination therapy. So hopefully next time we'll be able to give you some more data on this. But that's it, so thank you.